No? Yes. All right.
So let me just introduce our final speaker in the formulation and delivery session is Volker Schellenberger, who is the CSO at Amunix and has been working there for a few years on the XTEN molecule. And I first met Volker a few years ago when he was first looking at the physical chemical properties of these peptide extenders of half-life. So we're looking forward to hearing an update. Thank you.
Now I realize that I'm in a dangerous spot because I think I'm the only one standing right now between you and the poster session where there will be beer. So I'll try to be brief and I'll skip through some of the slides fairly quickly. Some of these data are actually on our website, some are published, and so these ones I'll skip fairly quickly and I'll focus on really the new data that you have not seen and you have no chance to see anywhere else at this point in time. So I want to actually, first of all, thank the organizers because I attended the same meeting last year. And attending this particular meeting last year really had a profound impact on the strategy of our company. And so I'm really grateful that I get a chance to present here and kind of tell you how this story continued on XTEN. And hopefully this time you will find that this is actually much more appealing to chemists because we heard when Wolfgang did his check in the morning when I presented last year, it was mostly for geneticists. And we know there are a lot more chemists in the room here. So this time there will be much more for chemists, which should be interesting. So just very briefly to give you an outline, so I want to introduce you very briefly to the concept of XTEN, go through some of the XTEN properties, which is biochemistry, half-life, safety, immunogenicity. I'll give you a very brief update on where our clinical candidates are, and I really would like to focus on this part, which is XTEN for chemical conjugation, because that's the new part where basically I want to tell you that we have now XTEN available for chemical coupling. I'll show you a bunch of coupling chemistries. I want to emphasize the homogeneity of the conjugates that we can make, and I'll, in the end, finish off with one of the more ambitious projects which also demonstrates what chemically can be done, which is a compound which has a peptide, XTEN, and the drug molecule all in one. So this slide really summarizes the idea behind Amunix that we have been pursuing now for 7 years. So if you look at PEG, it's really just a long hydrophilic polymer. And if you look at the protein, it's also a long polymer. But of course, we all know that they have very different properties because your normal protein will fold into a tight three-dimensional structure. It can be recognized by an immune system. So there are a lot of things which make a polypeptide behave very different from PEG. So our idea was, being protein engineers, can we come up with a particular sequence of amino acids which would actually make a protein behave just like PEG? So we were actually, typically the chemists tried to mimic the biology. In this case, actually, the biologists, we tried to mimic the chemistry because we liked a lot about PEG and we wanted to mimic that with the protein chain. So XTEN is really a protein mimic of PEG. And so why would you want to do that? I mean, first of all, a protein is composed of all natural amino acids, so it will be degradable just like any other protein. So there's no risk of it building up anywhere in the body. And if it does get metabolized, you'll just wind up with normal, regular amino acids. So there's no risk of building up any toxic metabolites. And the other benefit is that, of course, a protein you can encode by DNA. So you can actually control every monomer in this chain. So you have precise control over the sequence, the length. You can actually spike the sequence with specific amino acids for conjugation. And you can make this chain in large scale by microbial fermentation. So this is really what the presentation was, the focus was last year. So and this kind of outlines the process. You have a plasmid which encodes the XTEN sequence, and typically we have a protein which is either peptide or protein as a gene fused to it. So you transform that into bacteria, also in mammalian cells, and the ribosome will translate the whole thing into a protein chain. Your protein can fold into its normal three-dimensional structure, and the XTEN, as I say, has been engineered in a way that it does not fold into a three-dimensional structure. So the cell makes you a ready-made conjugate because these are linked by a normal peptide bond, and so you wind up with a molecule which looks very much like a pegylated peptide or protein. The limitation of that is, of course, that this is really limited to proteins, peptides, anything which you can build out of natural amino acids. And so when I gave that presentation last year, actually Rodney Lacks from Polypeptide Group came after me and said, I mean, this is all great, but I'm not a geneticist. My partners, the companies that come to me, they have chemical compounds. So is there some way, we like your polymer, is there some way you can give this to me that I can chemically conjugate it? And we knew that basically on paper this would all work, but we didn't know how well it would work and actually reduce this to practice. So this is basically the update that I want to give you. And I mean, on paper, this looks very simple. You just leave out your drug part. Now you make the cell mix just XTEN alone. And you can, again, you can conjugate either to the amino group. So that's if you just want to have a single functionalization. You can also have internal groups, lysine, cysteines, and you can conjugate in the middle. And so now you can, in addition to peptides and proteins, you can also include now peptidomimetics, organic molecules. So that's really, in a nutshell, the change that we have made over the last year, and I will really try to, because most of our biological examples are, of course, for these molecules, because we have just a lot more experience. But as you probably will see, it's the very same polymer that we're using, so whatever we learn from here, most of that should be transferable to the biological properties of this. And then I'll focus on kind of what can we chemically do to now get other structures made. So this is kind of another summary of comparing XTEN to PEG, which is really the mainstay, of course, for a long half-life polymer. So I already talked about the natural amino acids. I don't want to repeat that. But one other point is that because we are making this by a genetic process, we actually produce the XTEN as a single molecular species, while if you buy a high molecular weight PEG, you typically are dealing with more than 100 compounds. And that has consequences. So we can actually test our molecules, analyze them by mass spec, as I'll show you shortly. And that is very problematic for PEG-related compounds. And also, actually, our compounds are, because of they are perfectly homogeneous, we can actually purify them quite easily by preparative HPLC, which is much more problematic for PEG-related compounds. And in addition to that, we have actually a wider range of customization. We can cover a wider range. We have actually pushed it all the way past 100 kilodalton. So this is actually, these are not little peptides. These are really big proteins that we are making. And of course, we have this precise control of conjugation sites. This is a very brief summary of the properties. So if you look at the sequence, it's really just a bunch of hydrophilic amino acids. So all the amino acids which could be chemically unstable have been omitted from that sequence. If you look by mass spec, this is what it looks like. So it's really, there's really just one species that the mass spec can pick up. If you run a CD spectrum, it looks very much like a denatured protein, and this is done under physiological conditions, physiological and pH and temperature. So even under normal conditions, this sequence is denatured. And if you run a normal XTEN through a size exclusion column, it's the very first one that will come out of that column. So if these are all calibrators, this is an XTEN fused actually to a very short peptide. And if you calibrate the column, its apparent molecular weight is a little larger than 1 megadalton. So this is behaving like a very large molecule. And I don't have time to show you the evidence, but this is not an aggregate. So this column actually could resolve even larger molecules. Actually, we can make, if you want, in some cases, based on the payload, we can make dimers, and this column can separate these two. So we know this is, again, this is not an aggregate, but it's very, very large, as it was designed to do. So in the meantime, we have also been able to accumulate a very large number of PK data. And so they really fall into two categories. There are the well-behaved payloads, which are all the ones which are not subject to any kind of specific clearance mechanism. And they seem to all fall onto one trajectory. So if you go from between different species, as you go to larger species, the half-life goes up. And if you do allometric scaling, you actually predict a half-life of about 10 days in people. And then, of course, if you go to compounds which have some active clearance, then all bets are off. So then it really depends on the mechanism. So here I'm showing you just one example of the XTEN, because that's a molecule I'll refer to later again, and really it has been the benchmark through the whole day today. Almost every talk mentioned it, so where we have 130 hours predicted. And that's actually now also confirmed. Now, we talked about safety. So what is the problem with, what's one of the problems with PEG? Why is the lack of biodegradability such a problem? Because PEG is a very non-toxic compound. The problem really starts if you conjugate PEG to a peptide or protein, because sooner or later they will wind up inside of a cell. That can be the kidney epithelium, but it can be in other tissue if you have a receptor for your drug there. And so your protein part will of course be degraded, but the PEG is not degraded, and so it starts building up. And I think that is something that is more and more being appreciated, and very little is published about it, because usually whenever this happens in a clinical trial, the drug just disappears from the pipeline of the company. So there's very little that you read published, but pretty much all the large pharma companies we talk to admit that, yes, this is a real problem. And so if you have the XTEN, which is a protein sequence, of course, then you expect this to be degraded, just like the drug part of your molecule. And here I'm showing you just some gels which show that what happens if you take an XTEN fusion protein and incubate it in plasma. So you see that over a week there's very little change here. If you actually retrieve a sample even after an in vivo study, this is from a PK study, you see it after a week, the protein is still full length. If you take the very same protein and you incubate it with tissue homogenate, so now it's actually encountering proteases that are only inside of the cell, it's after in 1 day, it's all shredded into pieces. So that basically shows you that this protein is stable when it's outside of the cell, but once it gets inside of a cell, it's rapidly degraded, which is exactly what we wanted to achieve. Immunogenicity is a long story, and that's everyone is asking, I mean, how can you, this is a foreign sequence, how can you know that it's not going to be immunogenic? And the short answer is, I mean, I think only a clinical trial will ultimately convince everyone. But I think there's some reasoning why we even try to do this. So if you go through any of these epitope detection algorithms, there's really absolutely nothing. And the reason is that there is not a single hydrophobic residue in this whole sequence. And you typically, for T-cell epitopes, you need at least that one first residue, and then you need actually more than that. So if you run it also through any of these epitope and epivax prediction algorithms, and I'll show you one later, then it scores absolutely 0. We have done numerous immunogenicity studies in animals where we tested in mice, rats, rabbits, cynos, actually also dogs, and we have seen no evidence of immunogenicity. And of course, there again, it's the stories you will hear that, yeah, animal studies are not predictive of immunogenicity. That is true for drugs which are human proteins, because if you take a human protein, put it in a mouse, it's going to be foreign, the mouse is going to make an antibody, and you don't know what that means for a patient trial. The XTEN is really different, because the XTEN is foreign to all these species. It's equally foreign to a mouse, a rat, a rabbit, and a cyno. So all these species, all these mammals that we have tested cannot mount an immune response to it, and that gives us kind of the confidence that it's also not going to be immunogenic in people. So far, we have now 2 clinical trials where we have seen no evidence of immunogenicity, but these were all single injection doses, so I think the proof is still out there. Although, due to the long half-life, there was enough exposure to see an early onset immune response, but still, ultimately, only multi-dose, I think, will convince everyone. We have also not seen immunogenicity when we, even for its adjuvants, you can actually get antibodies, by the way, to the fusion partner, that you can, but you do not see antibodies to the XTEN. We actually were trying pretty desperately because we needed an antibody for PK studies. We have one, but it was really, really difficult. And that one, even that one is very, very weak. We also gave the sequence to Morphosis. They tried to they penned 4 times and gave up. They could not get an antibody to us. This is actually an epivax epitope analysis where you see an XTEN sequence, and this is a human protein. So the epivax, of course, does predict epitopes in the human protein. And of course, there the T cells would be removed in a patient, so that would not be immunogenic for very different reasons, because it's a human sequence and the human system has seen that. But in the XTEN sequence, the algorithm doesn't predict any epitopes to begin with. So even if the human system hasn't seen that sequence before, there aren't any T cells which recognize it. This is our pipeline, which for a company of 30 people is very, very ambitious. And so that list is probably quite a bit too long. And so part of that is, so there are 2 reasons why we can actually work on such a large number of compounds. One is that our discovery process is, of course, very straightforward. I mean, we make a fusion protein, and in the simplest cases, at least, we're done. There are some which are much more complicated than that. But for some of our lead molecules, actually, it was one fusion, and you have your lead molecule. After 2 months, it can go into animal studies, and you're ready to go. That's one component, and the other component is that we actually really like to partner our molecules very early, because we completely realize that we don't have the resources to understand the different biologies for all these different drugs. So, and the benefit of our technology is that we can actually apply to so many different targets. So we like to partner very early on with partners who understand the biology better than we'll ever do, and give it the molecule into their hands. So the most advanced molecules based on the technology are, I mean, that one was already mentioned, this is an XTEN4, which just completed phase 1, that's licensed to Diartis. We also have a human growth hormone licensed to Versartis. We have a very, very nice collaboration going on with Biogen Eidig on all the clotting factors. So this is actually proteins that we're making in mammalian cells. We're working on factors 7, 8, and 9 with them. We have another deal with a large pharma company which is not disclosed, and that one was actually just a few months ago was renewed because we kind of hit the goals of the first stage and now moving to the next one. So that's also going very well, very exciting biology. And then these are all internal programs, and I think the only thing I'll mention later on, I show a few slides about GLP2. So the same list is on our website. If this is interesting, we really like to partner early because we just simply do not have the resources to pursue all these molecules. So just a few data for the clinical studies. I think we have heard plenty about GLP-1 today, so I'll go very quickly. I think really the only thing I want to mention is, so this is a fusion protein. The GLP-1 is at the N-terminus, and we make this in E. coli. It's really, and actually maybe one more thing is important. The N-terminus was mentioned, is very important. So we produce this by a very specific protease cleavage site, and so which gives us a very clean processed N-terminus, and we co-express that protease in E. coli so that we don't have to purify a second protease. So that's part of how we built this production process. And just a brief summary. So of course, the molecule had to go through toxicity studies, and this was kind of nice because they pushed the doses very high. So mice have been dosed every other day, 50 mg per kg for 29 days, monkeys 35 mg per kg for 5 weeks, and there were no adverse effects tested. No antibodies against XTEN were tested in any of these studies. Phase 1 trial has been completed. The data have been reported. So this is just a very brief summary. So up to the highest dose, which is 200 mg, the drug was very well tolerated, very little nausea, which is typical for these drugs, as we have heard. There was a statistically significant reduction of hemoglobin A1C levels at 30 days. And also there was a dose proportional decrease in body weight over 30 days after a single injection of the drug. So certainly this looks quite nice. Now I want to go to growth hormone. Again, I will be very brief, but I want to pause a little bit more on this molecule because growth hormone is kind of the benchmark for all the different half-life technologies because there are 4 different products on the market. Combined, they actually have annual sales of $2.5 billion. And the situation has not changed in more than about 15 years. So these compounds have been all in parallel. And all the products are dosed daily because no one has come up with a good solution to allow longer dosing. There are now a few molecules moving forward. Actually, the most advanced one, at least with the best reported half-life, is a molecule that came from Ambrx. I think it's now with Merck. They reported in cynos 18 hours half-life. And so this, I'm showing you here the data which are from Versatis, where in monkeys, they actually reported half-life of 110 hours. And this is done by 3 different routes of administration. So A, we have record-breaking half-life, certainly for this class of molecules. And also we have excellent bioavailability. Again, a very brief summary of their clinical program. So they had, again, extensive preclinical tox where they saw no adverse event. They saw no antibodies XTEN. In this case, and that's actually known for growth hormone, you do see antibodies against growth hormone because it is a foreign protein to monkeys. They will make a response, but no antibodies to XTEN. Then they went into a phase 1 study in hormone-deficient adult patients. The result is that the molecule was very well tolerated. They had a sustained IGF-1, normalized IGF-1 levels for over 30 days after a single injection. And so currently, based on that, that's a very good biomarker for growth hormone. This seems to be the only growth hormone out there which has the potential for monthly dosing in this growth hormone, which is a very competitive space. Very briefly, I want to talk about GLP-2. So another peptide kind of structurally similar to GLP-1. So I just want to summarize very briefly the molecule. This is a molecule which we still have with inside Amunix, and actually we are quite interested in partnering this out. So we have actually done pharmacokinetics on it. The prediction, so it's 120 hours half-life in cynos, and its scaling predicts about 240 hours in people. We have clearly shown in vivo models that it has intestinotrophic effects, which is one of the indications. We have also run extensive multiple studies in a Crohn's model where we have seen increased length of the intestine, and we have seen improvements on a number of disease scores much better than the GLP-2 alone. So I just want to show you a few data here. So this is actually a Crohn's model where you basically insult the animal with indomethacin, and then you test, so this is the healthy animal control. If you treat them, so their intestine length is actually reduced. This is the free GLP-2, which is dosed twice a day. And this is an equimolar dose of the XTEN, and this was the very first study, so you don't want to kind of go too far out yet. So we did only daily dosing for this one, and we saw already a statistically significant effect. In fact, this one basically maxed out. It restored this marker back to normal. We have more, so this is a score where same animal model there, but now that the study was actually a little bit, the design was changed to kind of leave more room for improvement. So this is improvement of body weight, where you can see this is the control, this is the peptide alone, again, twice a day, and this is XTEN dose daily, and you see a statistically significant effect. It's actually better at equimolar dose than the free peptide, and that's because of this longer and continuous exposure. More data like that, this is actually reduction of TNF alpha, which is a marker, of course, for inflammation. And again, you can see that the XTEN with the daily dose, in this case in rats, actually does much better than the peptide dose twice a day. And of course, this much better than the control. Both of them work. I think I'll skip. So now I want to actually move. I have about 10 minutes left to talk about chemical conjugation to XTEN. So let's talk first about how we make this molecule. So I already said we are producing XTEN in a high-efficient process in E. coli. We actually get quite high titers of it. And E. coli makes, any recombinant system basically makes polymers, makes a protein at a much higher fidelity than a polymer chemist would make PEG. But it still isn't perfect. And so there's still cases where you have some molecules which are proteolytically cleaved, and also sometimes the ribosome just doesn't make it all the way through the mRNA. So you have some truncation products. And our goal actually was, can we push this to a situation where actually we can make XTEN at the purity level that you would expect for any other chemical that you use to produce your drug. And so in order to achieve that, we actually fused short tags to both sides of this molecule. And these are peptide tags which were optimized not for any kind of antibody binding. So they are binding to very cheap bulk resins that you can easily use and scale at a CMO and in a GMP process. In that way, we can actually purify this XTEN precursor based on these two tags very simply. And so only molecules which have really both ends complete make it through the process. We cleave these tags off with trypsin, which you can actually buy at GMP grade. And you are binding up with just free XTEN. And so this is a scalable process. So it gives us really high purity, as you'll see. And so right now, of course, we are doing this at lab scale, kind of 1 to 10 gram batches, which the process is still fairly expensive. But we have talked to CMOs, actually, including the ones who have made the clinical lots of our fusion proteins. And so based on their projections, we expect that once it's scaled up, we will have prices similar to polyethylene glycol. So this kind of drives home the point of purity. And so what I'm showing you on this side is that's Neulasta. So this is GCSF pegylated with PEG20. And so each of these peaks here, that's a different molecule. So each of these is a different length of PEG that you're getting. There's more than 100 compounds in here. And so even the most abundant one is less than 5% of the total mixture. And so this is an XTEN conjugate, chemical conjugate after purification. So the mass spec can detect only one species in this. And so just imagine what happens if you have a product like this, if you have any modification to your payload, if you have any chemical modification where now all these peaks will split into doublets or triplets. I mean, this is just very, very difficult to see, while here we can very nicely monitor, of course, what happens to the product. So one of the nice things about XTEN is that we can really customize it, so it comes in different lengths. We have a number of different conjugation chemistries, but right now we are really focusing on amino groups and thiol groups. We can have either one conjugation site or multiple conjugation sites, and we have also cases where actually we exploit the fact that we can have two different conjugation chemistries for orthogonal coupling. We have enabled a few different coupling chemistries. So this is a case where you actually have your peptide as a maleimide, and the XTEN has a thiol group, so you can conjugate that. And we have actually just recently shown that the same thing works with iodoacetamide, which gives you a more stable bond. So it's for research discovery stage, actually the maleimide is typically preferred, but for the development candidate, iodoacetamide gives you the more stable linkers. You can also flip this arrangement where you have an amino group on XTEN, you derivatize it with a cross-linker, and now you come in with a peptide or any kind of payload which has a free cysteine. We have also developed a cross-linker system for click chemistry. This is actually more for proteins, and this is at the moment only paper chemistry. We have not enabled this part where we actually convert the XTEN into an aldehyde. But all the other ones have been enabled, and I'll show examples of that. This is actually the first case. So this shows you really what a peptide coupling to XTEN looks like. So this is the free XTEN. You can run it by HPLC, get a nice peak. You can run it by mass spec. You know exactly that it's the right compound. There's not a single amino acid missing, nothing here. Then you add a very short, in this case, a maleimide crosslinker. So this is, just I want to drive home that point. It's a very small crosslinker you add to a protein sequence of almost 900 amino acids. And you do see a shift by a reverse phase HPLC. So that gives you an idea kind of how hydrophilic the XTEN sequence actually is. So the only thing which really sticks to this column is this little maleimide residue, which is sticking at the end. So you see you have a shift. You can quantitate how the direction went, and you can run the whole mixture through the mass spec, and it will tell you that, yes, I added exactly 218 Dalton to the mass, which is what you wanted. Then you, as a next step, you add your peptide, and you see, of course, now a much larger shift, and you also see an equivalent shift by mass spec. So you not only know that you did something, but you know you did the right thing. And actually, for these kind of reactions, what we typically do, we do them actually in one pot. We don't clean up in between because we can easily isolate these proteins after both steps by reverse phase HPLC. this is a case where we actually try to see how we can manipulate the valency, and this can be particularly interesting for compounds where the potency is kind of limiting and viscosity is limiting dose. So our reasoning was, XTEN, like any long polymer, is viscous. It's kind of in a similar range to PEG. And so our idea was we can, to some degree at least, alleviate that problem by adding multiple drugs to one XTEN. So what you see here is so basically the payload in this case is a peptide which has a maleimide. So we either use an XTEN which has one cysteine, and you see by mass spec that you're adding 4,000 something Dalton. If you take the same intermediate and you incubate it with this XTEN which has 3 thiol groups, now you're adding the peak shifts by 12 kilodalton. So you can very rapidly from the same intermediate go to multiple different valencies. This is a PK study where we actually compared, in this case for GLP-2, the recombinant fusion and the chemical conjugation, and this is at the C-terminus where basically there's not, which is not functionally relevant, and so these data are nearly superimposable, and this small difference actually is not statistically significant, and this is just what happens if you have a small company and you cannot afford to run 10 animals in each study. So there's no difference. So for the last few minutes, I just want to show you one example which is kind of a little bit more ambitious, but it shows you kind of what chemistry you can do with XTEN. So this is a class of molecule which has the following architectures. You have a long XTEN which kind of adds like the polymer backbone, and then you add a number of different cancer therapeutics to this molecule, and you also add peptides to the same molecule which will drive receptor binding and receptor internalization. And what you see, so if this protein gets internalized into a cancer cell, as I showed you, the XTEN is naturally biodegradable once it gets into the endosome. So this molecule will fall into pieces and you release your drug. So that's kind of something that comes natural to the XTEN. And of course, you can change the valency. So we're starting with a 3x3, but the expectation is that ultimately you can check how many of these targeting peptides do I really need, how many drug molecules do I really need to be practical. 3 is just a nice number. And actually, I'll show you in the next slide why that is. But no, actually, before that, of course, this area of targeted delivery of drugs to tumors is now very, very hot. And the main strategy how this is, of course, being pursued is this, is antibody-drug conjugates, where you take cancer drugs and you conjugate them to a tumor-specific antibody. So how does our technology compare with that? So both of these compounds will have long half-life. You would expect that. But actually, it is well known that the uptake of molecules into tumors is very much driven by their size. And so because it's not just tumor uptake, it's also uptake into normal tissues. So here we actually have the first advantage because you can actually customize this to maximize your therapeutic window as well. This compound, of course, comes in only one size. The next one is that antibody-drug conjugates actually are subject to FcRn recycling, which is the last thing you want to have for such a compound, because once you got it into a cancer cell, actually the FcRn receptor will take antibodies back out, and you don't want your drug to come back out. This molecule, of course, has a much simpler biology, no recycling. Another problem is hydrophobic drugs. Actually, just in the previous talk, we heard about paclitaxel. It's one of the most commonly used chemotherapeutic drugs. It is not used for this in this format because it's too hydrophobic. It makes antibodies aggregate if you conjugate it. We are currently working on a paclitaxel conjugate, and that looks very promising. Just from all our knowledge we have for extended hydrophilicity, we do not expect any solubility problems for even having multiple paclitaxels. We can have very good control for the number of conjugation sites, which is very challenging. And a lot of companies are working on this. It's very challenging in this format. Different manufacturing process, you may not care. But so this is another thing that these compounds after conjugation are very, very difficult to purify. Pretty much, they are just purified by sizing, where you remove the unconjugated drugs. While our XTEN conjugates are actually equally soluble in water and also in organic solvents. So that's why we can purify them by reverse phase HPLC. So we expect to have much cleaner compounds. I'll show you that. Aggregation, a lot less of an issue here. And as I say, these ones are always mixtures. Actually, they look pretty horrible if you look at the few published data for these kind of compounds. Well, I'll show you how clean we can make a molecule with this kind of architecture. So how do we make this? And again, this is a fairly complicated one. You can see more easy architectures, but I wanted to show this kind of really to show what the toolkit can do. So in this case, we're actually building this compound from 2 different segments. They are both based on the same XTEN. So we're starting with an XTEN where we actually use the alpha amino group, and we have 3 thiol groups for adding either the peptide or the drug. So we make one segment which has the peptide and has 1/2 of the click chemistry at the alpha amino group. And this can be done in a one-pot reaction in one day, HPLC purified. You do the same thing here. You add your drug to the cysteines, you add the azide to the amino group, purify that by HPLC, You mix these two compounds together, they click together, and you have a compound which looks like this. Here you see the HPLC. This is one segment. This is the drug segment, which is a lot more hydrophobic. If you mix them together, you see there's a little bit left over of the peptide segment. There's a little bit of leftover here of the drug segment, but you have this large new peak, which is the conjugate that you want. And you can purify that by HPLC. And after that, it looks like this by HPLC. So it's really, as far as we can tell, it's a perfectly pure compound. If you run a mass spec, it has exactly the mass, which is XTEN plus 3 peptides plus 3 drug molecules. If you run it through a size exclusion, it runs just like any XTEN would. So these little peptides, drugs that are attached here don't affect it. It still runs at about a megadalton on a size exclusion column. So with that, actually, I wanted to say we're getting actually a lot of interest of parties who want to try this. So we actually started to build a collection of these XTENs, and this is expanding quite rapidly, where we manufacture these at gram scale and offer them under a fairly simple material transfer agreement, so that people, if they say, I'm not trusting this XTEN stuff yet, you can basically get these reagents, try it in your own, run them side to side to peg and see whether you like the result. And so we can make a lot more basically on a custom basis if needed. So in summary, just want to very briefly say that we have a polymer which is composed of natural amino acids. It's encoded by DNA. It's extremely hydrophilic, has a very large hydrodynamic radius. It has a chemically defined structure. We can do a lot of different formats. It's very flexible. And 2 compounds have been in clinical trials. And with that, actually, I just want to thank the team that has really done all this kind of work over 7 years. It has been a phenomenal, really, ride to get through all this. And I'd like to thank you for your attention.
That's a terrific update for the last year. I have a quick question. What is the longest half-life biopolymer that you've made?
I can only tell you about what we have confirmed data for simple fusion proteins in cynos, where we have gone to about 7 days. But we have not really pushed that to the limit. And the problem is it was done GFP, and because the methods get better and your half-lives get better every time. And so we basically, when we got this data, we realized that we should really should have run the in-life study longer than we did. So all this PK modeling hinges pretty much on the last data point. And so you cannot quite know, maybe it was 10 days, maybe it was 6 days. You can't quite be sure. So I wish I have basically another program where we can push this out and run it for a month in the cyno study and know how long we can really go.
Very nice. I'm sure there's a question over here.
Oh, sorry. So you mentioned that once these things get into the cell, intracellularly, but the proteases degrade them very effectively. The examples you showed, though, they were all basically staying in circulation, so they're staying extracellularly. So how do these, I'm confused, how do these clear ultimately? Are they ultimately taken up by liver cells, or?
That is our expectation. Actually, we have done, from one of the earlier versions, we actually have done a biodistribution study with radio-labeled XTEN, and that looked like it was mainly in a central compartment, ultimately clearing through the liver. Even that early one, which wasn't as large as the ones we now have, there was nothing going through the kidney.
Okay, so you've done some radioactive studies where you measure content in bile and feces, etc., or?
We did, I have to say, it was a fairly crude study, but you could certainly tell that most of it was cleared similar to what you would expect for a large molecular weight PEG.
Do you have different molecular weight like the PEG, 20,000, 30,000?
Oh yeah, no, we
Not
go.
One fit all. No.
So no, absolutely not. So we have, the most favored one right now, which is in a clinical trial, is 864 residues.
Yes.
But actually, the growth hormone actually the story is actually, the paper will be published, I think, in a month or two, where you'll see that this story was very complicated because there's receptor-made clearance involved, and actually this molecule has 2 XTENs, a short one and a long one, to get that kind of long half-life. And so, but now actually, we can make the XTEN even longer than that. And the main boundary was what we had learned is that, initially we just simply didn't know how to express such such odd sequences. So there wasn't really a limitation. Otherwise, we simply couldn't make them. And now we can make these long sequences in multi-gram quantities per liter. And so now actually we can comfortably go longer than even our favorite one. And we simply didn't have time to do the biology to follow up because we are focused on all these lead compounds that we have. There's just only so many things you can do in a small company.
I was not listening to the answer, what you told him. What was the half-life? 10 days, you said?
It's somewhere around 7 days. That's where basically I'm comfortable to say. Again, I'm hoping we can go longer, but the PK data, it's just the statistics isn't good enough to really prove that.
Good.
Yes, Volker, nice work. These conjugations are second-order reactions, so I'm just wondering, do you have to dose the ligand at high concentrations to drive the chemistry of conjugation, or can you do it at one-to-one stoichiometry? Because these things are presumably occluded as they...
Yeah. Actually, there is not much occlusion, so because this is still a very hydrophilic polymer, so small molecules have no trouble accessing it. But you have, of course, that problem that your molar concentration of the XTEN is not going to be very high simply due to its high molecular weight. And so it depends on the chemistry. I mean, if you use NHS chemistry to the amino group, you're going to have a large excess because you'll lose a lot of that reagent to hydrolysis. If you use the maleimide chemistry, that's fairly clean, so you need only about a 2x excess, even a little less than that. If you use the click chemistry, that actually works really well. And so we pretty much do the reaction one-to-one. Because you just have no competing hydrolysis reactions which eat your reagents.
For the click reaction, what catalysts and additives are you using?
This was, I mean, as you know, there are different versions. This is actually one of the copper-free versions. So you just, because, but I don't think this will be our clinical molecule. This is just a very rapid route to testing which molecule works, because we don't expect it, because this click bond is buried in the middle of XTEN, so we do not expect it to affect biology. But I think ultimately, when you make the molecule, I think you'll go a different route.
Right, I was just curious. You know, there's a lot of amines present, obviously. Is there a leaching problem? I mean, I'm assuming PrEP HPLC would get rid of that, but have you looked into that?
You can say about the leaching?
Leaching of copper. So in other words, copper binding on to the peptide.
Again, we are not using, we're using copper-free click.
Oh, I'm sorry. I thought you said you, okay, copper-free.
No, we use copper-free click at this point.
All right, thank you.
Yeah. So it's beautiful. You just mix two compounds and you can go.
Hi, I had one question about the fusion proteins that you expressed. You said it could be done in mammalian cells as well as E. coli. So the sequence, it flashed up a bit too quickly for me, but it looks a bit like a mucin. It's very serine, threonine, proline rich, which is ideal for Olink glycosylation. So would you not have you run XTEN sequence through like NetO-glyc? You
do see significant glycosylation if you express it in mammalian cells, yes. And so then, of course, also this benefit that you have of having a single molecular species goes away. But the reasons we are focusing there very much on the clotting factors for these with the Biogen collaboration, because there actually the challenge is these are very complicated molecules anyway. They're anyway glycosylated, and the big challenge is to do any kind of site-specific kind of connection to a polymer. Yeah, factor VIII in particular is a quite large molecule, so doing specific modifications on that one is challenging.
Well, let's thank Volker again for a terrific talk.